Hello everyone, my name is Dr. Gary Ho. I'm a neurologist and physician scientist at Brigham and Women's Hospital, and I'm very grateful for the support of the APDA in supporting our research on new approaches to the study of alpha-synuclein dysfunction in Parkinson's disease. So what is alpha-synuclein? So alpha-synuclein is a normal neuronal protein that has functions in neuronal communication. However, in Parkinson's disease, alpha-synuclein becomes dysregulated, and this causes the alpha-synuclein to aggregate abnormally or form clumps. And over time, these clumps can develop into what's called Lewy bodies, shown here. And these Lewy bodies, under pathological conditions, are what define Parkinson's disease. Right now, we have quite a few symptomatic treatments for Parkinson's disease, but we don't have any medications or drugs that target this underlying process that causes the disease. And for this reason, our research is focused on trying to understand what happens to alpha-synuclein when it becomes dysregulated and how we can potentially compensate for that deficit. So our approach that was supported by the APDA to try and address some of these questions is to use these induced pluripotent stem cell-derived neurons. And what that involves is taking skin cells from either Parkinson's disease patients or persons without Parkinson's disease, and using various techniques, we can reprogram them in a petri dish into induced stem cells. And from these stem cells, we can, under specific conditions, differentiate them or turn them into neurons, brain cells. And using these brain cells from Parkinson's disease patients or those without Parkinson's disease, we can study the function of alpha-synuclein and try and understand what goes wrong in Parkinson's disease. One of the things that we have been able to achieve is that using these induced pluripotent stem cell-derived neurons, we've been able to develop a new method to study how alpha-synuclein affects communication across neuronal synapses. This involves the chemical tagging of neurotransmitters and monitoring their transmission across two neurons as they're trying to communicate. And one thing that we have found is that in Parkinson's disease-derived neurons, the communication between these two cells is impaired, and alpha-synuclein, being dysregulated in Parkinson's disease, seems to inhibit the release of these neurotransmitters in a way that does not occur in normal neurons. Having understood this, we are now able to try and find different ways, different drugs and various compounds to try and restore that communication deficit in an attempt to try and alleviate some of the symptoms and, more importantly, to try and address some of the underlying dysfunction that is present in Parkinson's disease. One of our strategies that we've been looking into is a process known as palmitoylation. So palmitoylation is a modification of proteins by the fatty acid palmitic acid. And when proteins are modified by palmitoylation, it confers upon them certain properties that they did not have before the palmitoylation. One thing we found is that alpha-synuclein, when present or when dysregulated, causes vesicles, or these bubbles that contain the neurotransmitters, to be misshapen and to aggregate. And when we increase palmitoylation of other proteins that are also on the vesicles, we're actually able to normalize the structure and function of these vesicles in an attempt to try and restore neuronal communication. So this is one strategy that we've looked into, using a chemical compound that increases the levels of palmitoylation in these neurons. And we found that by increasing palmitoylation, we're able to restore a lot of the normal functions that were previously adversely affected by the dysregulated alpha-synuclein. Now, we've so far talked mostly about testing our concepts and testing our ideas in these cultured neurons. However, one thing that we've been also able to do in collaboration with some of my colleagues also at Brigham and Women's Hospital is to take some of those concepts and try to apply them in a different model, so a mouse model, which has the advantage of being able to test certain behavioral phenotypes since this is a whole animal. And these mice have certain mutations in alpha-synuclein that produce a Parkinson's disease-like symptoms in these mice. And one of those symptoms is a deficit in cognitive functions, in particular visual spatial cognitive functions that we can often see in Parkinson's disease dementia. So for example, the Parkinson's disease model mice, when they're put in this special maze called a Y maze, have impaired visual spatial reasoning. This Y maze is initially blocked off, so one out of the three arms is blocked off, and the mouse is allowed to explore the other two arms. When the barrier is removed, a new, what we call a novel arm, is now available to the mouse. Under normal circumstances, the mouse, being curious, is going to explore the novel arm and spend more time there than the other two arms. However, in the Parkinson's disease mice that we've been using, that is impaired, and so they don't really recall whether or not one arm is a novel arm or a familiar arm, and they end up exploring all three arms roughly the same amount. And we've been able to use that compound that enhances palmitoylation, apply that to the mouse, have the mouse take this drug over some period of time. And in collaboration with my colleagues, we've been able to show that that also improves their cognitive function as measured on this test. So that demonstrates that targeting palmitoylation can be one promising method to try and address the underlying disease pathology in Parkinson's disease in our ongoing efforts to try and find a disease-modifying drug to treat Parkinson's disease, all of which was made possible by the support of the APDA.